see these different steps. You reacted at all these different temperatures. You have to have it at 5 degrees, then cool it to -10 and -15, and then -50 degrees here. And then you run these reactions. This one's done at 130 degrees. This one's done at 60 degrees. You're like, what's with the temperature? What's your problem? Why you're cold, hot? Why do you do this? Oh, because we just like to. We just like to cool things down and heat things. No, you have to. Each step needs its own parameters. How does nature know that? You say, well, it was, it's some deep sea vent. Okay. So it warmed up there. And then what? And then it left the vent. And then it, ended up at some other really cold place. And then went back. And then it went back and back and forth. Nobody knows how any of this ever happened in nature. Nobody knows. Next slide. So this is the experimental procedure. So what you do, you take an oven-dried 3-neck round bottom flask, and you charge it with 33. You add magnesium sulfate and dichloromethane, and you cool it down here, cool it down. This is just one step on that thing. And then you get the yield. This is just one step. There's all these things you got to do. You have to know what you're doing. Even if I just gave this to you, you wouldn't know what to do. So you have to be an experienced chemist to take this and to be able to repeat this. So what is a mindless abiological system? What does it know how to do? Nobody knows. Next slide. Then you have to characterize it. Once you make what you want, you have to characterize it. So we have this tool called NMR, and you get these peaks. And from these peaks, you assign the structure so you can get the exact structure. This is how all the pharmaceuticals that you take, this is how the structures are assigned. You run these spectra. And you say, well, nature doesn't have to do this. It doesn't. Nature uses a more complex system. It has a different glove for every hand in nature, a different enzyme that detects structure problems. It's more complex than this. This is a general tool. But the next slide shows us how, next slide, this is what we have to write to convince our colleagues that we got what we thought we got. A combination of standard deuterium, proton, C13, depth 135, C13 experiments with standard 2D, H1 cozy, H1 NOE, boom, boom, boom. So you go all through this. Not done. Next slide. So that's part 2. All of that needed to just characterize the one molecule. Characterization is harder than synthesis. It really is. Any chemist will tell you. You can make something, but it takes you a lot longer to characterize. And you have to know the structure because you have to know what to do for the next step. Nature is confronted with the same thing all the time. Making molecules is hard. Next slide. This paper had 281 pages of supplemental characterization data. I showed you a paragraph of characterization, 281 pages of that to convince the world that we made what we were making when we made those light-driven motorized nanocars. How does a dumb abiological world ever do this? Nobody knows. They're all clueless, but scientists aren't saying how hard it is. Next slide. This was the first generation motor that we put in the car. It worked, but it only rotated at 1.8 revolutions per hour. When we rebuild it without that sulfur atom there, then that 5-member ring, then it goes 3 million rotations per second. All right, so small changes can make a big difference. Well, what do you do? Well, what you do is you just come here and you erase that sulfur and you do that. Now that you can do on a blackboard. But this took us back to step 1. This is what I'm talking about. When something, you're going along and you want to build and you say, oh, I'm just near life. If you have one thing wrong, it doesn't work. So, well, you just modify this and it'll work. All right, go back to the beginning. Go back 1 billion years to the beginning. It's too hard. Nobody knows how this thing is solved. You can't build the molecules. Next slide. And then, so remember, these just to make the molecules, the simple molecules, you got to have the carbohydrates, you have to have the lipids, you have to have the nucleic acids, you have to have the proteins. Even if you make an amino acid, how do you hook the amino acids together? Amino acids don't hook together by themselves. We're not even talking about order. How do you just get them to hook? I mean, somebody sent me a paper just last night. I mean, here's a paper just came out on self-folding molecules. Well, duh, that doesn't prove anything. You build a molecule, it folds up. Big deal. How do you get these things to hook together in the first place? Nobody knows. That's with amino acids. How about with nucleotides? You want to get these things to hook up. People will make you see, we got a nucleotide. We got DNA. No, you don't have DNA. You got to get these nucleotides to hook up. Look at a DNA synthesizer. How many steps of blocking, protection, deprotection you need to do the complex chemistry just to get one linkage made? Nobody knows how. You can throw all these together in a flash. People say, well, the concentration of nucleotides was very high. I've read this in high school books. It was very high. And these things came together. You calculate the concentration, the world would have to be raining nucleotides. And even if it were raining nucleotides at those concentrations, they wouldn't hook up. They don't hook up. You need enzymes to hook them up. But this is prebiotic. There's no enzymes here. Or you need very complex DNA synthesizers. Those weren't there either on early Earth. Now you have to assemble it. So say we had the molecules, how can you get them to assemble? So here's the assembly experiments. A protocell is a self-organized, endogenously ordered spherical collection of lipids proposed as a stepping stone to origin of life. So that's what a protocell is. But most so-called protocell assembly experiments in origin of life research can be summed up by a protocol analogous to this. You purchase homochiral diacetyl lipids from a chemical company, or you synthesize stereoscramble lipids from smaller molecules. Add those lipids to water and observe a small amount of it to form simple and expected thermodynamically driven assembly of those lipids into synthetic bilayer vesicles upon agitation. Sometimes the researchers will add other molecules that get engulfed by those vesicles. Then you publish a paper claiming that the synthetic vesicle is a protocell and suggestive of early forms of life. Engage with the media, hype it up, watch the layperson be misled. Every assembly experiment, I'm telling you, can be summarized in this. Whatever you send me, don't send me anymore. I've dealt with it all right? I get enough of them. And people say, well, what about this one? What about this one? And it fits all into this. It's all garbage, all garbage. Next slide.